Hey there. Today I attended the lecture of The Physics of Genes and the Promise of Personalized Medicine. It was given on Thursday, November 17th, and the speaker was a biophysicist from the University of Illinois. So the topic of this lecture was really about how biology and physics have large interplay and how you can determine different key aspects about how DNA compacts and functions within cells by looking at their charges and relationship between the actual atoms within the DNA. So to start off, this lecture really got down to the biology of physics, which for me was very exciting because I'm a biology major and physics doesn't really super make sense to me most of the time, but I really enjoyed this lecture. So the lecturer started off by showing us what DNA was composed of, which is the 4 base pairs, adenine, guanine, cytosine, and thymine, and then how they stack together in hydrogen bond to make a double helix molecule. So DNA is really just one molecule, and all those atoms come together to make a complex structure. The big thing he mentioned with that DNA structure is that the atoms have different charges. So like the phosphodiester bond on the DNA backbone is very negatively charged, and it's interesting that the DNA molecules can be compacted so well because you would think that with such a strong negative charge there would be a lot of repulsion there. But the biggest point he made, talking about DNA, was that charges repel in general. So if you have like 2-point charges, both of the same charge, they'll repel because likes repel and opposites attract. But if you have those charges in a media, as DNA is in the cell, in the cytosol, which is a lot of water and ions like sodium and chloride, then those strong like charges can actually become attractive. So he used an example of a big bag of salt versus a big bag of road de-icer, which is also a salt, but I guess with a stronger concentration, or like more ions, stronger charge, I guess. So in that comparison, if you add just regular table salt, you get 2 DNA molecules and the salt coming together, and it's not enough for them to overcome that repulsion, so the 2 strands would still repel and compaction would successfully occur. But if you have something like magnesium chloride, which is in the road de-icer, then the 2+ charge of that is actually enough to give a positive charge to each of the DNA molecules at, say, 1 base location, and then you can actually have an attractive force between those 2 molecules, which I found to be quite interesting, because I've looked at the compaction of DNA from a biological standpoint, but never thought about it in terms of the physics and why it is actually possible to get such a long molecule, say about 2 meters of DNA, into such a tiny space that is the nucleus, which is only about 10 nanometers in diameter. The other things he talked about were how his research and other researchers have been able to use, again, the physics of the DNA to expose some really cool components that led to both the sequencing of the human genome and then later development of DNA origami, which is a really cool new method of using specific molecules that you design to attach to the DNA to bring certain segments closer together in order to fold the DNA into different shapes to provide different mechanisms for the cell. So if, for example, he had one that was like a box with a lid, and so you could put some sort of drug in there, like a cell-targeted drug, like a cancer drug, into that tiny DNA box, and then if you put antigens on the lid of the box, they'll recognize the cancer cell, bind, and then open the lid and release the drug into the cell for a very targeted and personalized approach to battling some of the toughest diseases that we're facing currently as a society. Let's see, what else? I think one of the coolest things that I learned was really about the charge relationship and thinking about it in a way that's different than pure physics and different than what biology has taught me. Biology doesn't entirely skip over the physics, but it definitely doesn't go into as much depth as we did in this lecture, which was really cool. And the other really cool thing that he showed us is that his team is working on a device that actually can sequence DNA by pulling DNA through a biological pore, in this case a protein, based on its charge. So again, looking at the physics in order to sequence and get down to the biology of the DNA, which is really important for sequencing and bringing the cost of sequencing down, which again will lead to more personalized approaches for medicine and drug delivery. So very, very cool stuff. I think overall the lecture was very interesting, especially relating to all the biologists who are in the audience, but then also to those who weren't biologists, he still made it very, very clear what he was doing and described everything in a way that I think even if you didn't have a background in the subject, you could still understand what was happening. A couple of things I researched since the lecture were that more DNA origami shapes. So he showed us the box, and he also showed us one that his team was working on, which was something very similar to like a syringe. So you could take your DNA origami, bring it to a cell, and then inject your drug target or whatever you want to into the cell. And I thought that was really, really cool, especially seeing as I want to go into medicine in the future. So I researched more into this DNA origami, and it turns out you can really make any shape you please. You have to first design the shape and then identify which molecules you would need to target your specific DNA sequence to kind of make the DNA conform to the shape you want. But in doing so, you can do all sorts of really cool things. A team at Harvard that I looked up actually was making a robot to kill cancer. So again, using the principles of DNA origami, they were able to make a similar sort of box, but put like tiny nanoparticles. I don't really understand robotics, but anyways, they could use the DNA origami concept so long as pH is stable and things of that condition to deliver tiny targeting robots to specific cells, specific cancer cells or specific tumor cells, which would then release them safe and sound into their desired target where they could act and destroy and do everything that they need to do to get rid of that cancer. So that was probably the coolest thing, and the thing that sparked my interest was all the DNA origami and the drug delivery applications. So that's what I've been looking into since, and I really think there's a huge future in that, especially since sequencing and things are getting so much easier. We'll be able to look at a person, look at whatever ailment they have by looking at their DNA, and then make really, really specific approaches to how to defeat and treat those illnesses. So that's really cool, and it's definitely going to be within our lifetimes, I believe. Hopefully I can be a part of that someday with research and whatnot. So here's looking forward to that. Yeah. So all in all, really good lecture, really informative. I really liked how he made everything really easy to understand, and how he related physics to biology, which brings in some of my favorite topics and all the importance of genetics into a whole different perspective. So, cool lecture. I recommend everyone look into DNA origami and personalized medicine applications as a result of it. And yeah, that's all I got to say.